Hello and welcome to our first video blog of the Microbiome Power. And we are very honored and happy to announce our today's guest, Mr. Ross Youngs, a serial entrepreneur and the CEO of the Biosortia Microbiomics. I'm also very excited to announce that this is going to be the first video blog of Microbiome Power, and credits go in large part to Mr. Youngs. Because he encouraged me to do the video instead of just doing standard blog interview. So first I'm going to say good morning, although it's morning in Ohio and afternoon in Heidelberg, Germany. And good morning, Mr. Youngs, and how are you doing?
Good morning, and I'm doing very well. Thank you very much.
Great, great. Nice to hear that. Your company, Mr. Youngs, is as well as our company are dedicated to the same goal, and this is the application of the microbiomes and the microbiome science for the benefit of the humanity of our whole planet. The technology that I read about and that you patented and which you are developing is really astonishing and is really promising to radically change microbiome mining. But before we dive in deeper into the details, I wanted to say I've seen a couple of your previous TED Talks and podcasts, and your career path looks very exciting and unorthodox, I would say, for someone in life science business and especially in microbiomics. So maybe first I would like to ask you if you could tell us a few words about yourself and your career, and how come that you became dedicated to microbiomes and microbiomics, and actually how Biosortia was born?
Terrific. Well, thank you again. I appreciate the questions and I appreciate the opportunity. And I'm a generalist. So when you look at how people approach things, they can approach things as a specialist or as a generalist. I've had a lot of exposure to a wide-ranging sets of technologies, sciences, engineering, and I think that that's worked well for me. And I think for others, they have been able to dig into scientific expertise and become the experts in their fields and really focus. Both are needed in this world. As a generalist, you're right, I have created other companies, other products, other technologies, but I've never been more excited about a technology and an opportunity than I am around microbiome and microbiome mining. I get goosebumps when I actually start to think about what this is potentially going to do for humanity. So it's really exciting to be involved in this. I have to go back to 1975 when I took my first class in microbiology. And here I am decades later, hopefully leading a revolution in mining the microbiome, not only for the genomics, but for the actual hidden chemistry and the activity of that chemistry, and then being able to connect the dots. So this is an exciting time, and I think my entire background, the diversity of backgrounds, allowed me to get to this opportunity, create this technology.
Wonderful, wonderful. It's nice to hear that, because we are excited as well as you probably with the microbiomics. That's why we started Microbiome Power in the first place, in order to connect people and to raise the awareness about microbiomes. So let's talk maybe a little bit about Biosortia technology itself. It could be a game changer globally in the fields of microbiomics and beyond that. And this technology is promising to harvest small molecules from microbiomes, and it might have a huge spectrum of potential applications, from drug discovery to agriculture, etc. Could you maybe shortly elaborate on the principle of how this technology works, and what are the advantages of this technology to the other microbiome mining tools?
That'd be terrific. I definitely will talk about how the technology works, but I think just a little bit of a lead in as to why. Why was this technology needed? And I think we have to go back to the 50s. And in the 1950s and the 1960s, the vast majority of new products coming from pharma and agriculture were directly related to microbes. And the issue there is you can grow microbes in the lab, scale them up in bioreactors or other growth systems, and ultimately obtain enough of a microbe that you get to study its hidden chemistry. Problem, less than 1% of the microbes on the planet can grow in a lab. So all of science, since the point they shifted from growing microbes in the lab, and obviously lots of researchers are still doing that today, but with such limited access, that's created issue. And when you only have it growing in an isolated culture, it's not interacting with the other species in a microbiome. So metabolic pathways aren't turned on. And if you study it indirectly, which is what virtually everybody in the world is doing, genomics, synthetic biology, computational biology, AI drug discovery, all of those approaches are indirect. They don't go explore the molecules nature has made through billions of years of evolution. So the challenge is, why didn't they go look at natural microbes? And I would say they did. And they ran into a couple very big problems. One, microbes change their metabolic pathways in a matter of minutes. So you can't typically take a snapshot of a living microbiome and lock the chemistry in place that's being produced, because by the time you harvest it, take it out of its natural habitat, things change. The other issue is the diversity of microbes, their sizes, their fragility, their ability to be able to withstand the pressures of harvesting, or some of the other technologies, which would be chemical processing, can destroy cells in the process. So you end up collecting old material and the wrong material. Our technology actually is designed specifically for going to natural microbiomes, taking a massive quantity at a massive quality out, giving us a high quality starting material in what works out to hundreds of kilograms dry weight microbiome. We have one collection that's actually 1.4 tons of dry weight microbiome from a natural habitat. And the whole goal there is to be able to dig deep into these species that no one's ever been able to study and find their chemistry. And the whole objective with something like that is to not only explore the chemistry and the activity of the chemistry, but the connections to the genomics, so that we can build those AI models with real data, and at the same time collect molecules that could be starting points in either drug discovery, agricultural, cosmetics, sustainability. The whole life science realm will be disrupted by having direct access versus just indirect access.
Mhm. Yeah. I see. I see. So I saw actually you're mostly focused on the aquatic sources. Basically, you talked about processing the aquatic microbiomes. Why is that? And are you also considering other possible sources of samples instead of aquatic?
That's a great question. Why we like the aquatic environment is that is essentially when you go to a particular site, you have a large quantity of microbiome available. And it's homogeneous to a certain degree when you get into a certain habitat. And you can choose your habitat in the aquatic environment. The variabilities are immense. So think about the world as a natural culture and all these different habitats, whether it's temperature, whether it's pH, whether it's nutrient or the types of nutrients, whether it's oxygen content, whether it's pressure. All of these variables exist in different places. And what we've found, or I shouldn't say what we found, what science has found, and the Terra Oceans Project was one of the ones that really helped understand this, is the diversity of the microbiomes across the aquatic environment is huge. Such an extent that even the marine microbiome overlaps our human gut microbiome by more than 73% in genes and metabolic pathways. So if you're trying to collect enough of a microbiome in order to be able to study its hidden chemistry, you want to start with something that's relative to all microbiomes. And the aquatic environment, because of evolution, because of its starting point for all of life, is a brilliant starting point. To your question about could there be other ways to use this technology to collect microbiomes? And the answer to that is absolutely. So if we wanted to look on the terrestrial environment, let's say soil, we look at a way to use our technology to collect microbiomes from those habitats as well. If you talk about plants, we're looking and exploring at all sorts of possibilities to scale microbiomes to a quantity and quality that we can explore them so that we can actually impact humanity in a very positive way. And yes, we're a for-profit company, so we want to do this and return investment dollars to those that have invested in us as well.
Sure. Yeah, that sounds like a very, very wise decision, to go to the aquatic microbiomes in the first place. Another thing which actually I noticed is a broad spectrum of applications, and actually this is how in the first place I became a great fan of your LinkedIn posts, because they are so diverse, and we really also like to look at the broader picture, and the microbiome power as a company, we would like to bring a broader overview and to raise awareness not only about human microbiomes or the human gut, but also about the animal microbiomes, about plants, about the environmental, about all possible microbiomes. And I'd like to brainstorm a little bit with you about possible applications of these technologies. You mentioned in some of your talks and the differences between the traditional approaches in, for example, drug discovery, between the Biosortia and the traditional pharmaceutical industry drug discovery approach. Maybe you can tell us a little bit about this, and also maybe about your vision of other applications in animal or in the veterinary field or in the agricultural field and other possible fields.
Terrific. What I look at, what our scientists that work with us, what our collaborators look at, is they first look at the small molecules inside of microbes as brilliant starting points. Some of these are actually good drugs, good natural drugs, all on their own, but they become the molecular starting point for something that can be improved or optimized. If you start with something that already works cell to cell, like a microbe, you're starting with a better, let's say, structure to move into biology. If you start with something completely synthetic, it's never worked in biology. You may have a lot more work. So when you talk about starting points, the ability to advance something that comes from microbes into these various applications, whether it's drugs, whether it's an agricultural product, or whether it's a sustainability product, is improved automatically. I've had researchers, in fact, very smart people talk to me about, we have all the molecules we need. We have more than enough molecules to search through. And I find that a little naive for someone that has spent their life looking at molecules, like this one individual did. And that's because when you start with something that's completely synthetic, you've got a huge disadvantage. when you start with something that's natural, you've got a huge advantage. And it reduces the early discovery cost dramatically. And I think that that's one of the biggest points of digging into what is really a limited pool of actual natural small molecules throughout nature. Someone could say, yeah, that may be billions. It may be billions, but a billion, is a relatively small number when you talk about all of the synthetic possibilities, which would be, potentially 100 orders of magnitude above a billion. So you've got to start with what works and build on what works and improve what works. And that's really our focus. And I think there's a number of people that recognize what nature has done through 4.2 billion years of evolution are the best starting points.
Exactly. That sounds fantastic, really. And we are all looking forward to see these future applications. But however, at the end of the day, we, as you mentioned, also we also are looking to generate some profits, and it will be certainly interesting also for potential clients and also for potential investors or the other interested parties to discuss a little bit about your business model. I've heard you saying that basically you're focused on selling the platform technology, not doing the development part. And could you maybe shortly explain about Biosortia potential revenue streams and, for example, selling, licensing of the technology, and who would be or who might be the potential customers?
No, that's fantastic to ask that. The way we look at it is if we remain focused on discovery, utilizing our technologies that we've invented, we've created, and we've proven, we're in a position to participate in a number of projects that move forward, potentially hundreds of projects that move forward. So Biosortia itself wants to remain that early discovery company and will probably take things as far as IND in time. At that point, we want to hand it off. Now, we may hand it off to a pharma company we've got an arrangement with, an agrochemical company we have an arrangement with. It may become a NewCo through an academic collaboration. And that would be great because our model is to license early so that we can move a molecule quickly into optimization to IND. We'd rather license early or license it when it does become an IND. And I like that model personally because when I've gone around and talked to pharma companies and pharma VCs, every group out there is clearly focused on buying at just before IND or at IND. They want to pay whatever they need to pay to get a de-risk molecule. Biosortia will be a machine for creating de-risk molecules. Unlike a biotech, which really picks a molecule or a series of molecules around a specific interest, their success and failure depends upon them to get into development, where they can end up seeing ultimately higher valuation for their company and then an exit. They're working on one project typically. We could be working on 200 very diverse pipeline molecules across a dozen different fields. And our risk is dramatically reduced. If a molecule makes it to phase 1 and it has to back up or it gets delayed, that will not crush our company in any way whatsoever. We'll have dozens of projects moving forward in a situation like that. So as a business person, I want to de-risk my company as well. And when you have one product you're staking your entire future on, that's scary. I'd rather stake it on 20 or 30 and leverage my collaborators, my partners, those that are paying me to find these molecules, turn them over to them early, then get those milestones and those royalty payments as they become products. That creates a company that is looking and thinking long-term and making sure that it stays on top when it comes to discovery. And we want to be the best at finding these molecules from nature and then creating that starting point that can turn into patents and IP that others can leverage.
Okay, so these are actually, you also mentioned you're creating the libraries, right, of the small molecules, and then out of these libraries, some of the molecules are those that you mentioned that are in these pipelines, these 200 pipelines, for example. This is one thing. So basically, you're also going to sell the access to the databases for the molecules and the libraries, right?
That's exactly right. So one of the things that will happen from one recovery, if we decide to go to a specific habitat and collect that microbiome, we do that through a series of initial analysis. Once we've made that determination, we collect a massive amount of material so we do not run into a, let's say, a stop because we can't get enough material to pull out the molecule that's active. So we've eliminated that issue right from the front. But with that diversity of chemistry we get, we can see typically 70,000 small molecules at the scale where we can put them in well plates for others to test. Now, that's one microbiome harvest, and that 70,000 small molecules will probably in one event alone be more small molecules from microbes that the whole world's ever tested. That's what's so amazing about what we're doing, is our ability to dig into these molecules. And when we look at our own data, quite frankly, what we see is a large portion of that has never been tested. So, researchers have discovered and identified this molecule, but they never had enough quantity to ever test in assays to understand what it does. In this case, our library will consist of novel molecules no one's ever seen. In fact, there'll be at least 15% where no one has ever even seen these molecules. And then there will be a larger percentage than that where no one has ever tested these molecules. And this is from one harvest. We hope to be in a situation, 3 years from now, where we're doing a harvest a week somewhere in the world, and then have the processes in the system set up to mine these microbiomes so thoroughly and so rapidly, we get at those novel molecules, put them in the well plates for testing, for assaying, so that we can start to understand what this molecule does. That all contributes to data, whether it's the habitat data where these microbes grow, the species data, all the genomics, all the metabolic pathways, all the gene clusters that exist within all of this, then the actual chemistry, and then the activity of that chemistry. This is a database that does not exist.
This is fantastic.
They're in pieces.
This is really fantastic, really. Like basically you're covering the whole chain. I also saw you're selling the harvesting systems, and you also mentioned servicing metagenomics. Could you please explain how exactly you are servicing metagenomics?
Yeah, so our goal in our microbiome mining factory will have a genomics component to it. So the metagenomic side of it, the ability to understand the microbes is just a piece of it. The ability to understand a species, its genes, its metabolic pathways will be critically important in connecting the dots. So we want to make sure that we're across that spectrum with that. The other part of your question, I'm sorry, I got so stuck into the metagenomic side of it. You had another part to that question.
Okay, okay, good. I just wanted to ask you another thing because since our company is also focused on providing networking services for all parties active in the microbiomics and the microbiome science, uh, either academic partners or the so corporate partners, and I was thinking about to which possible kinds of other collaborations or partnerships you would be open. For example, I was thinking about let's say metabolomics because you're also dealing with the small molecules, and there should be certain overlaps. Or for example, your technology is generating huge amounts of data, and then could you imagine maybe a collaboration with another big data management or a platform or a software or artificial intelligence or similar? And would that be interesting for you, or basically you are tending to keep all of that in-house, so to say?
Yeah, no, our whole goal is to be highly collaborative. So our opportunity to work with other people that have their databases for finding mutual opportunities, we'd like to find a win-win. And we'd like to be able to do that. If the win-win is they pay us for access to portions of our database, and then they find things that help them advance, then that's a win-win. If we do it as a collaborator and mutually try to discover something together, that's a win. And then I remember the other portion of your question that I wasn't responsive to, and that was the technology, the sale or licensing of the technology itself.
Mhm.
So we're a big believer in collaborating and expanding. So we do hope to be able to license our technology into other areas, whether that be other countries. We'd love to be able to do that because what will happen is we build a powerhouse of data collection. And what we've seen already is when we do a microbiome recovery over here, we learn certain things. When we do it over here, we learn things that are different, but sometimes it's the same. So one of the molecules that we were, let's say, very excited about as a possibility, we ended up finding it in one harvest at 1x. We found it in another harvest at 7x. And it was because we identified it in the first harvest, we were able to search for it in other harvests and find it. So we could see the future where, let's say, microbiome mining using our technology platform and our approach is occurring in Asia somewhere. They find molecules and are looking for more of something. And because we're collaborators, we can say, yes, we found that. We've got more of it in stock. If you guys need it for screening, let us know. We can ship it to you. And the same thing with the data. We want to see this explode globally, obviously for a opportunity to benefit Biosortia, but we want to see this explode globally so microbiome mining becomes one of the most important tools for understanding the hidden secrets of cell communication.
Exactly, exactly. I completely agree with that. And we are also tending to look globally, although we are based in Europe, but we tend to look globally because actually this is the only way how we can reach the benefit for everybody. And just a little bit more about the business. Your funding. I've seen you've had support from ARPA, from DARPA, from U.S. Air Force. These are already credentials that tell enough about the level of the technology. And what are your plans, or what would be your message to potential investors and plans for other supporters of your efforts?
As far as we've gone, which is really kind of interesting, between the non-dilute funding from those agencies and then the dilute funding, we've put about $22 million into this technology so far. We're completely scaled at the harvesting, so we can go out and do a harvest whenever we want. And we're proven on the harvesting leading to the molecules of interest and potential. So everything about the technology is proven. What we need to do is build that microbiome mining factory. And that factory itself is essentially a biorefinery that it takes our raw material, which is lyophilized microbiome at scale, moves it through that highly efficiently and effectively so we can slice and dice it and get it, not only the lipids, but also the small molecules and other valuable components of the microbiome itself. That we're going to do our first A round, and we don't know exactly how we're going to do that. So we're exploring it, trying to figure that one out. And our goal is to execute at scale for the first time. Everything we've done up to this point has been at subscale. And all of that subscale research has led us to believe that we need to be able to go to a scale. That scale will be on orders of magnitude beyond where anyone has been, including ourselves, to this date. So if someone can explore a liter of water to understand the microbes that's in there, to understand the initial chemistry in there, imagine exploring 20 million liters of that same source.
Yes.
You're going orders of magnitude into the chemistry that is really important. And that's the chemistry that's at nano and picomolar concentrations that really is the most potent chemically signaling chemistry that we typically can't even begin to see or understand. So we're just going to go very deep and learn things that are relative to all of life sciences.
I see. So it's basically also about upscaling the whole thing. Yeah, fantastic.
Yeah, definitely. Because some of the issues that people have run into is they get a hit, they go back to their source, and they don't find that hit again.
Yeah.
They didn't collect enough to begin with, or it's transient. And microbiomes are always going through succession. They're always changing. Species are being added, species are being eliminated. And it's all that communication, that defense, that warfare, that repair, everything that goes on there is what's so critical to life everywhere.
Sure, definitely. Okay, that's basically that's basically that we are now approaching the end of our talk, and maybe the last part. I would like to ask you about the future. So, what is your vision for the future? I mean, a part of it you already answered now through the other questions. I wanted to ask you about your plans for spreading around the outside of US and so on, but you already answered that. So maybe some other vision, because you're visionary, some vision about the future developments of the technology and the application of your platform.
Yeah, I guess because we've all have been impacted by the health and welfare of the people around us. Like my mother passed away from leukemia. The hope is that small molecules come out of nature, and especially microbes, because that's really what we're investigating, that can impact disease and impact suffering. That's first and foremost what I really hope we get to do. The second thing that I think is critically important revolves around sustainability. So when we look at these secrets of life, can they end up changing a dynamic with toxic algal blooms? Can you turn waters that are non-productive and not healthy for the residents that live around them or use them into healthy waters? And I believe there's the potential of that occurring. Could we reverse coral bleaching? Could we help capture more carbon? So sustainability is one of those issues.
Definitely.
And then the last is food. Can we find the secrets of the microbiome that improve productivity, that improve the range of how crops could grow in arid conditions or in challenging conditions?
Exactly.
So when you talk about a vision, it's broad And any one of those is a career pursuit. And it won't be me. It'll be the fact we've brought these molecules to researchers that are pursuing those things. Can we find a cure? Can we find a better herbicide? Can we find something for sustainability? And it's going to be in those partnerships. We're just a tool to hopefully facilitate that. And I would love to be able to execute this tool at scale.
Sounds fantastic. This is exactly what we are also looking for, like to have the complete picture of people doing the omics technologies on one side and people doing microbiome mining like you do. That's really exactly the direction I believe the whole branch should go into. And good. In the end, I think my final message would be actually to all people potentially interested in this great platform and this great technology, and all people basically interested in microbiomics and beyond, whether they are researchers or a company or an investor, please contact Biosortia directly, or you can also contact us at microbiomepower.com, and we will be delighted to help you. And finally, I would like to thank you, Mr. Ross Youngs, for your time and for a great discussion. And I wish you a lot of success in the future. And I'm looking forward to our future collaborations. Thank you very much.
Terrific. I as well. Thank you so much, Vladimir. You have a great day, and thank you for this opportunity.
Thanks to you too. Goodbye. Have a nice day. Bye-bye.